I'm Dr. Mila Brujic, and today on the OI podcast, we have Dr. Maria Walker from the University of Houston, where she's going to be unfogging fogging.
Friends, this is Dave Kading. Before we get into the show, I wanted to thank Team for supporting the podcast. If you're not aware, Team is a virtual staffing and agency which helps connect virtual teams to your practice. I don't know about you, but I have a harder and harder time finding people to come in and join me in the in-office staff. And so over the last 2 years, we've found 10 virtual people to become part of our team. They do things like answer the phone, they help with billing insurance, they check for documentation before it comes in, they scribe in the exam room, they can order contact lenses, and the list goes on. We've been able to work with several agencies over the years, but we have found that Team has been the most effective at ensuring that our virtual employees get paid, stay on time, and that we're following up with them and making sure that they're a great agency for these people. If you are ever interested in considering a virtual employee into your office, I'd love to chat with you. But also, I wanted to let you know that Team is offering a $250 discount when you first sign up with them. Check out hireteam.com, hireteem.com for a $250 discount, or see the show notes below for a link that will get you to the website. Thanks to Team for supporting the OI show, and thanks to you for being a listener.
Dr. Maria Walker, it's always a pleasure to have you on the show. Welcome, welcome, welcome. So first and foremost, please, if you could tell the audience a little bit more about yourself, where you practice, what you spend most of your day doing.
Sure. Hi, thank you for having me. Yeah, I'm Maria Walker. I am in Houston, Texas. I teach at the University of Houston College of Optometry. I've been here for about 10 years. I came here after I did my residency in contact lens. I did a PhD here looking at scleral lenses and how they impact the eye and the ocular surface. And I've been on full-time faculty for almost 3 years now. And so my days, I see patients a couple of mornings per week, mostly scleral lens patients, mostly keratoconus. And then the rest of my time is spent in the lab or writing, teaching. I teach the contact lens course here at the University of Houston, the intro to contact lenses. And I've got a lab, I've got PhD students, many masters and postdoc and stuff like that. So
That's awesome,
kind of
Maria.
split it in a few different ways.
Yeah. So you're all things contact lenses then. I think that's a testament to the individuals that you worked with in your residency as well, too. I've known you again since you were a resident, and you've always had this undeniable passion around specialty lenses. But boy, for somebody to predict that scleral lenses would be such an important topic today. I mean, it's really overwhelmed us in terms of the patients that we care for and the way that they can provide better vision, and also just the literature and the amount that's coming out. And you're one of the people that are actually adding a lot to our understanding about this. But you're unique because you're looking at the tear chemistry within the scleral lens, within that post-lens tear reservoir. And you're finding some interesting things about the foggers, the midday foggers. And tell the audience a little bit about kind of what you're working on and what you're finding.
Sure, yeah. So I've always been interested in the scleral lens research. And really, since my residency, I was very interested in this fogging and this accumulation of some sort of debris in the fluid reservoir and what it actually is. And of course, how do we get rid of it? How do we figure out who this is happening to? So I always have at least one of the projects that I have going on is always going to have to do with fogging. So we have a couple right now, but one of the things that we're about to publish on actually is, so we've looked at what's actually in it. And a couple of years ago, we published on there being lipids in the fluid reservoir in midday fogging. And we've also shown in unpublished data that there's somewhat of an increase in some of the inflammatory cytokines. So like the proteins that basically are driving inflammation everywhere in the body.
So
We also,
Maria, excuse me, you're taking these lenses off and you're
yeah.
actually testing the fluid that's in that lens. Just out of curiosity, like
Yeah.
do patients have to take, do patients literally have to take them off, like down, just to make sure that they're not like dropping any of the fluid out?
Yeah, so we remove the lenses. So we actually have a pretty well-established protocol at this point that has been evolving over the past 10 years. And so what we do, we always remove the lenses for the patient. I have had one patient who really didn't want us removing it, and she actually did a very good job at removing it herself. But essentially what I do is I have the patients like stand up and they'll bring their head parallel to the ground. They'll hold their lower lid. I carefully remove the lens. Um, and then we kind of like let the fluid sort of drip into the lens from that. And so we end up, and then we'll do a little rinse. So we'll rinse the lens with about 25 microliters, depending on the study, saline. And so then what we basically have is like 50 microliters, and 1,000 microliters is a milliliter. So a very small amount of fluid. And what we do with that, we know that there's cells in there, there's debris in there, and then there's what we call soluble right? So things that are dissolved in the fluid. So we actually separate those out. We centrifuge the samples. Then you basically get like all the cells and the heavy stuff will go to the bottom. We remove the top called the supernatant. That has all the dissolved proteins and dissolved lipids. So then we can separate those out and we can look separately at the things that are dissolved in the fluid and the things that are whole, like a cell, or, sometimes you just get just debris, but it almost always ends up being either an immune cell or an epithelial cell, which are very easy to distinguish because epithelial cells are very big and amorphous, and immune cells are very small and have specific morphology. So that's sort of how we've
Maria,
developed
when you
it, and
say
we can
epithelial
test.
cells, are they corneal epithelial cells or conjunctival epithelial cells or both?
Good question. We think that they're corneal epithelial cells, but we actually, that's a good point because we haven't actually we haven't actually done like antibody staining to confirm, but based on the morphology, like put it this way, I've never seen a goblet cell. That's a really easy cell to kind of tell what it is. The epithelial cells, as it gets to the surface, they become much thinner and more amorphous.
Hmm.
And so when we see those, we think that they look like sort of the surface superficial epithelial cells morphologically. But yeah, I suppose you're right. We could have some conjunctival epithelial cells in there. But I don't know enough about their specific morphology. We haven't stained for them specifically, but no, that's a good question. They appear to be a corneal epithelial cell. They just look like what we see when we do corneal epithelial cells in the lab. And we know
So
that those are corneal.
you're finding some interesting things though between these foggers and these non-foggers. Like what are some of the differences that you're finding between these two patients?
Yeah, so first thing is that there's definitely more cells. And again, the epithelial cells are present to varying degrees, but what we see pretty much in every patient that has fogging is an increase in immune cells, and specifically an increase in neutrophils, which neutrophils are the most common. They make up about 60 to 70% of all the white blood cells that we have in the body. And they're what we call part of the innate immune system, right? They're first responders. So they'll respond to anything. And what we think is happening is that as a lens lands on the eye, applies some mechanical pressure to the blood vessels that are in the conjunctival tissue. And what happens when any blood vessel is mechanically stimulated is it will dilate and release these cells because it doesn't know what the insult is, but there's some insult. And so it will send out the neutrophils to sort of basically gather some information about what is the actual insult. And then those will then send other signals to recruit other types of immune cells, depending on, right? Because if it's a bacteria, for example, right? You need a very specific response to that. In this case, it appears to just be mechanical. And so sometimes I find myself wondering, these cells are probably so confused, right? Because they get out to that ocular surface, they're trapped in that fluid reservoir. they don't appear to be necessarily sending a signal back that says, all cells come here, we have an insult, we need to attack it. So it's almost like maybe they get released because of the vasodilation, and then they just sort of are there because there's not a whole lot for them to respond to. There's not typically a bacterial infection, although, you know, as we know, there's lots of, you know, there's what we call a microbiome just naturally on the ocular surface anyway.
Sure.
So in some ways, these might be good because these are phagocytic cells, right? So if there is bacteria, we know there's, even if there's not an infection, we know there's going to be bacteria in the lens, right? There's bacteria on our hands, there's bacteria everywhere. So you fill a lens, you put it in, there's going to be bacteria there. Now, is it going to rise to the level of overwhelming our system and creating infection? No, not often. So sometimes I wonder if maybe these midday fogging patients, they get the cells there, and then it's actually protective because maybe they kind of are gobbling up just the little bacteria that are running around naturally. And I mean, everybody knows on the eyelids, we've got all sorts of bacteria and thing. So yeah, they probably are eating up some of those. And actually we've looked a little bit at that because you can look with very high resolution. So like transmission electron, like the highest resolution imaging basically possible. And you can see bacteria in the neutrophils, but we've looked at that in other scenarios, but not in these midday fogging. So that's a good, you're going to give me all these study ideas. I guess we could do that in another project, but,
Look at this, you're just
yeah,
getting more and more stuff to do. Maria, you're just like total job security here. So you guys are doing some real neat stuff though, where you're taking the fluid from the post-lens tear reservoir, then you're then introducing it to epithelial cells in a petri dish, correct?
Yes. So all the work we've done, one of the questions that my old lab manager actually had was, you know, it's hard to know everything that's in that fluid. And so we sort of had this approach where we're like testing for this, testing for that. But then we sort of thought, well, on the other side of that, can we just look and see if this fluid causes an inflammatory response? Because we can't test for everything necessarily, but one of the questions we wanted to answer is if an epithelial cell is in constant contact with this fluid, could it potentially be creating an inflammatory pathway? And so what we've done is we've got these collected fluid reservoir samples, and we basically have these dishes with very small little wells because our fluid volume is so low that we actually you can use just what we call cell culture and just take these cell lines of human corneal epithelial cells. So we take these cells, we let them sort of attach to the wall. So this is very common, very standard cell culture techniques where you have a type of cell, you kind of put it on a little petri dish, and then you can treat it with whatever you want. And so we plate these corneal epithelial cells from normal individuals. So we haven't progressed into disease epithelial cells, which could respond differently. And then ultimately, we'll put the fluid reservoir in culture with the corneal epithelial cells. And the treatment, we typically will treat them for about 30 minutes in an incubator. And then you can actually tag with antibodies one of the major transcription factors that essentially is like the light switch that turns on genes in that cell to then produce inflammatory cytokines. So the pathway we're looking at is called the NF-kappa B pathway. NF-kappa B is the most sort of ubiquitous. It's called a transcription factor. Basically, it's sitting in the cytoplasm of all cells. And if something insults that cell, it will then move into the nucleus and literally turn on gene transcription for interleukins, for MMP9, for basically all those inflammatory proteins and cytokines and proteases. So we can tag it with a green fluorescence protein. And what we've seen is we do in the midday fogging samples tend to see more of that green concentrated in the nucleus, which tells us that transcription factor what moved into the nucleus. So those cells are responding into inflammation in that way.
So it's interesting. So I love microbiology, and just in total transparency, I was a biochemistry minor in undergrad, so I love this stuff. So for the audience, when Maria's talking about transcription and translation, transcription is literally the process where a protein goes to the human DNA and it translates or transcribes, excuse me, a little strand of messenger mRNA. That messenger mRNA is then going to become a protein through translation. So those are just two different processes. But what she's talking about is when she takes this fluid and puts it into a healthy cell, you start to upregulate inflammatory proteins that will be produced by that cell. This is interesting, Maria. And the reason why I say that is because, so clinically, what I've seen, and this is anecdotal experience, when we take these patients that maybe have misaligned haptics and maybe have more of their weight bearing on a part of the sclera and conjunctiva, uh, again, in a very easy way to think about it, in a with the rule sclera, you have on a spherical landing zone, you have more of this weight that's bearing nasally and temporally. And it may be part of the reason why we tend to see fogging in those patients, and just simply putting a torque haptic on that landing zone reduces substantially, if not eliminates, what we see as fogging. And it may be because of this weight that you were talking about and the fact that we're now more evenly distributing it. So there's not heavy pounding of this weight on the nasal and temporal aspect like there was before. And maybe this more even distribution of weight is giving us less of a impetus towards this fogging. But you're literally conferring for me that not only is fogging bad because of the response that patients are actually seeing, it's going to make vision blurry, but we actually now need to be thinking about mitigating it or reducing it if possible, because there is something that's different with this than the standard non-fogger. And there is something that can potentially have an inflammatory role. We don't know it yet, but we know that there's something more to it than just disrupting the vision. Is that maybe a correct pull through on that clinically when I describe it that way?
Yeah, I think that's a fair assessment. I think in a lot of ways, midday fogging is a way of the eye telling us there's too much pressure on these blood vessels, or you're creating a response, because regardless of, we know that there's sort of different severities and different components. But the thing that does seem to be consistent with all patients is an increase in these neutrophils, these primary attack cells. And so, yeah, I think that this is the eye is telling us you've done something, whether or not it's mechanical stimulation of the blood vessels, which is what you and I both think is what's going on, hasn't quite been confirmed yet, but it's telling us this pressure is too much. You're causing us to respond by sending in our first line of attack against any insult, mechanical, bacterial, or otherwise. And yeah.
I think that's awesome, Maria. Well, listen, we've took up too much of your time today so far. We are so appreciative of you kind of sharing at this level, really what's happening, what's going on. I think when we're fitting specialty lens patients in particular, I think our understanding of clinically what we're seeing, but also doing a deeper dive and really understanding what the downstream ramifications may potentially be, just help us position this so that we're a little bit more cognizant of all of this as we go with our patients. So thank you for sharing this with us.
My pleasure. Always fun to come chat with you.
And thank you for unfogging the mystery of fogging with our scleral lens wearers. And all of you, thank you for joining us on this episode of the Optometric Insights Show. Cut! Woof, 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 woof. The dog pound! Woof, woof, woof,
Love that.
woof.
Oh my gosh, you're right.
Maria, you're so
That's
awesome.
fun.
That was great. That
Yeah,
was
thanks for having me. So fun.
Oh, thank you for being available, Melissa.
Yeah.
Now I keep calling you
Yeah.
Melissa. Maria. I called you that once by accident during the show too.
And
Oh,
now it's going to be like in your mind, you're going to keep calling me that. It's all right. I'll correct you every time. I don't mind. I guess I kind of look a little bit like a Melissa. Generic blonde
look,
white girl.
you don't look anything like a Melissa. Don't ever say that again.
I'm my full
Yeah.
unique Maria self. Put it this way, every time you call me Melissa, I'm going to start singing the Maria song. That will help keep the mnemonic for you.
Good. I'm like a Pavlovian dog. Like you just need to
That's
keep
right. That's right.
me when I do something wrong.
Slap you on the hand.
Well, Maria,
Yeah.
thank you. Thank you. Thank you. Have a great weekend. Have a great Easter, all that good stuff.
Yeah. Happy Easter to you guys. How's everything going? You got Easter plans?
Yeah. Yeah. So we're Serbian Orthodox. So we actually celebrate by the old
Oh,
calendar.
yeah.
So ours is
So you
May.
what?
We celebrate Easter on the old calendar. So ours is actually May 5th. Is when our Easter falls this year. So we're like Greek Orthodox. We're very similar. But saying that, we always celebrate both. Like we're going over to Sonia's family tomorrow for Easter, and then I'm sorry, Sunday, and then we're gonna see my family just tomorrow, just for a visit and everything. But yeah, it's gonna be a good
Oh,
weekend.
fun.
Yeah, and
That's great.
I got band practice tonight.
Oh, fun. What a full life. I love that. Well,
We
have
got
a
a
great Easter.
we got a show
Huh?
April 13th. Come to Bowling Green and see us.
That's amazing.
It's
Wait, when is it? April? Yeah.
April 3rd. It's Saturday, April 13th.
Gosh, what a
Yep.
riot. You
So
know what's
this,
funny? I'm actually, because I'm going to be there whenever the eclipse is next week, because I have to be at OSU for a meeting. So
oh,
I will be in town, but I don't think I'll be there for the rest. Yeah.
stay that weekend. Yeah. Is it the 8th? Monday, April 8th. You know, Maria, we're
Yeah.
actually closing our office at noon that day. Like this part of the country is like, we're gonna like
This is like the place to go for it. Yeah.
yeah, and
So
actually,
we were buying,
well, Columbus is okay,
our
but
ticket.
we're more in the path. Like Northwest
Oh,
Ohio is like more in the path of like total eclipse.
uh-huh.
They say there's going to be crazy people coming in for this thing.
Right. So I went to buy tickets to come in, and it was like $1,000 to fly to Columbus back. I don't care because it's a part of a clinical trial, so they're paying for it. But we were laughing because we were like, wow, I guess we're in the right place at the right time. Like we're in a good spot to see it. So I'm psyched.
So stay for the rest of the week and come up for this show on Saturday.
Oh God, I wish I could. I had to be back in the clinic on Tuesday.
Boo.
I know.
Wait a
I
second.
know.
So you're flying back.
Someday.
You're flying
Yeah,
back.
Monday night.
Oh,
Monday
okay.
night.
So you'll see the eclipse. All right. Well, good.
Yeah. Oh, I'm sure like we have to see it. Like we're going, yeah. And especially
Yeah.
at the optometry school, all the optics
Yeah.
nerds will be, they all like it's like an event.
Oh,
So
yeah.
it should be fun.
Good. Well, Maria, enjoy
Yeah.
in a week from now your time in Columbus as well
Yeah.
too.
Yeah.
All right.
Sounds good. See you soon,
Okay.
Milay. Thanks for having me.
See you, Maria. Thank
Bye.
you. Bye.
Thanks again, friends, for joining us for this episode. Again, thanks to Team for their support of this episode. If you're considering or have ever considered getting a virtual team member for your practice, check out hireteam.com. That's HIRETEEM.com. Mention the OI show when signing up, or hit the link below for a $250 discount off of your first month's team member.